Hello everyone. So we are going to discuss on the topic that is de novo synthesis of fatty acid. So this process is solely associated with the synthesis of fatty acid within the human body. For your better understanding, I have kept this topic in 3 sets of presentation, which comprises part 1, part 2 and part 3. So basically you should know that where it actually takes place. This synthesis basically takes place in different organs, which include the liver, the kidney, moreover the adipose tissue, and sometimes apart from that it also takes place in the mammary gland. Now what actually this process do? This process it do the simple genesis of fatty acid from carbohydrate or sometimes from the proteins or amino acid. So we are going in details from the part 1. So basically, for your basic introduction, the basic definition of this de novo synthesis, it is also known as the de novo lipogenesis. Lipogenesis means the genesis or the generation of lipid, which is a endogenous pathway. It generally produce the lipid about which I have already told you that it actually produced from the carbohydrate and sometimes from the proteins. Moreover, this process it takes place in the liver, in the kidney, in the adipose tissue, and also in the lactating mammary glands. Moreover, the third part you should remember before going to the depth of this process is that the enzymes which are responsible for the genesis of lipid are generally found in the cytosomal fraction of the cell. These enzymes, they are generally located in the cytosomal fraction. So this process it is also considered as extra mitochondrial because it takes place outside the mitochondria or cytoplasmic fatty acid synthase system. Now there are many products which are produced from the process of DNL, but basically in your body or in human body we used to get the palmitic acid, the palmitoleic acid, the cisvecinic acid, stearic acid, oleic acid, etc. Now again, if we see here, I am repeating that same thing only. Where it occurs, it basically occurs in the liver, in the adipose tissue, kidney, brain, lactating mammary glands, right? Now what is the substrate which will start the process of the lipogenesis? So basically the acetyl coenzyme A, it is the source of carbon. I repeat, the acetyl coenzyme A, it is the source of carbon for the genesis of the lipid. Moreover, the NADPH, it provides the reducing equivalence, and this NADPH it is generally produced during the process of HMP shunt as well as Melick enzyme reaction. So the reducing equivalent it will be produced during the process of the lipogenesis by the NADPH. Moreover, lastly, every molecule of acetyl coenzyme A, I repeat, every molecule of acetyl coenzyme A delivered to the cytoplasm. Whenever each molecule of acetyl coenzyme A will reach the cytoplasm, one molecule of NADPH will be formed. So basically the ATP it supplies the energy for the genesis of the lipid in your body. So basically you can see here in this particular diagram, so this is the process. Starting material, it is the acetyl coenzyme A. It will go polymerization, it will undergo polymerization to reach a 14 carbon chain or 14 carbon molecule. Then again it will go elongation to reach 16 carbon atom. Now actually these are 16, 1 and -1. Actually this is a particular nomenclature which is given to the fatty acid. So basically it is written as sometimes as 16 n minus x. So here this n, what this n represent? The n is nothing but it is the chain length. It is the chain length. This n, it is the chain length of the fatty acid. Whereas this x, actually here in the general nomenclature you will have one x. 16 is to n minus x. Here x, this x represent the number of carbon. I repeat, this x represent the number of carbon from the double bond to the terminal carbon atom. So what is the number? So this is the x. In generally we used to have, or we used to write like that, 16 is to within bracket n minus x. I already told you what is n. It is a number of the chain length, or it is the chain length. Moreover, here the x, it is a number of carbon from the double bond. Now the two basic things you should know. First of all, 7-hexadeca, or you can say 7-hexadecenoic acid. 7-hexadecenoic acid, it's another important fatty acid which is produced by this general method of de novo fatty acid synthesis with a partial beta oxidation process, right? Now this particular fat, this 7-hexadecenoic acid, this particular fatty acid, actually it is somehow related to your health system. It has been actually reported in different health related journals that these fatty acid, actually they could affect, or they could increase the risk of coronary heart disease, which is abbreviated as CHD, or sometimes it can raise the condition of sudden cardiac arrest, that is the SCA. Now stages, in what stages these fatty acids are produced? First of all, the first stage, it is the production of acetyl coenzyme A and NADPH. It is the first crucial and essential step for the production of the fatty acid. The second step, it is the conversion of acetyl coenzyme A into malonyl coenzyme A. And the third method, or the third step, it is the reaction of this fatty acid synthase complex. Now coming to the next part, so the production of acetyl coenzyme A and NADPH, so how it is going to take place. So first of all, you should know that this acetyl coenzyme A, it is the starting material. It is the starting raw material for the synthesis of the fatty acid. Moreover, this acetyl coenzyme A, it is produced, we have already come across, it is produced in the mitochondria by the oxidation of either your pyruvate or fatty acid, or sometimes degradation of carbon skeleton of certain amino acid, and also sometimes from the ketone bodies. And moreover, this mitochondria, you should know that this mitochondria, this is not permeable to the acetyl coenzyme A. So now the question is that we come to know that the acetyl coenzyme, it is the raw material, right? So this acetyl coenzyme A, now it's need to be utilized for the synthesis of the fatty acid, but the synthesis, it takes place outside the mitochondria. So how the transversion, or you can say transport of acetyl coenzyme A from the mitochondria to the cytoplasm, it will take place. So basically the conversion of acetyl coenzyme A to malonyl coenzyme A, it is considered as the prime method or process for the transport. How it takes place, you can see here, this part, it is the mitochondrial part, this is the mitochondrial membrane, whereas this is the cytoplasm. So what are the reactions those are taking place? So first of all, it's a alternate arrangement that is made by a cell, or it's a bypass arrangement to transfer the acetyl coenzyme A to the cytoplasm first of all. Now, what actually happens? This acetyl coenzyme A, it condenses with the oxaloacetate. It takes place within the mitochondria to form a third molecule. It is the citrate molecule. Now, this citrate, we know that it can move freely outside the mitochondria to the cytoplasm. Now, you see, again if we see here, again I am showing you, this is the mitochondria, this is the cytoplasm. So what are the reactions which are taking place? Again, in the cytosol. Now what will happen in the cytosol? So in the cytosol, it is cleaved by ATP citrate lyase, means the citrate, it will be cleaved by ATP citrate lyase to liberate again the acetyl coenzyme A, which has actually formed or which is actually present within the mitochondria. This has been converted into the citrate and it has been sent out of the mitochondria. Right now it will be cleaved by ATP citrate lyase to liberate the acetyl coenzyme A along with oxaloacetate. These are the two products, right? It is taking place outside the mitochondria in the cytoplasm. The next method is this oxaloacetate. This oxaloacetate in the cytoplasm, it will now get converted into the malate, right? The malic enzyme actually, it converts the malate to the pyruvate. So finally this malate, it will get converted into the pyruvate, and NADPH and carbon dioxide are generated in this reaction. See here, NADPH, it is produced. Moreover, when this malate it get converted into pyruvate, carbon dioxide, it is also produced. So both of these, this NADPH and carbon dioxide, they are utilized during the process of fatty acid synthesis. So here I am giving you the details pictures. So this is the mitochondrial matrix, the process which generally takes place within the mitochondria, and this is the cytosol. So how it generally takes place, how this process is generally takes place. So basically you should know that the transport of acetyl coenzyme A from the mitochondria to the cytosol, it is always coupled with the cytosomal production of NADPH. We have come to know that, and carbon dioxide, which is highly advantageous to the cell for optimum synthesis of the fatty acid. I repeat it once again, the production of NADPH, what we have shown in the earlier slide, and the carbon dioxide. So this makes the cell optimum, or it favors the cell for the synthesis of the fatty acids. Now, so up next, the process is the formation of malonyl coenzyme A. So this is the chart. You can see here how this malonyl coenzyme A, it is produced from the acetyl coenzyme A by the virtue of the enzyme acetyl coenzyme A carboxylase. So in brief, first of all, the acetyl coenzyme A, it is carboxylated to malonyl coenzyme A by the enzyme. It is known as the acetyl coenzyme A carboxylase. This is the enzyme. Moreover, this process, the conversion of acetyl coenzyme A to malonyl coenzyme A, it is a energy dependent pathway. So here we need the ATP. We need the ATP for the conversion. So it is a ATP dependent reaction, and it requires the biotin. It requires the biotin for the carbon dioxide fixation. So here you can see enzyme biotin, it requires the enzyme biotin for this carbon dioxide fixation. Moreover, this mechanism of action of acetyl coenzyme A carboxylase, the enzyme acetyl coenzyme A carboxylase, it is similar to that of the pyruvate carboxylase, which we have already discussed. And finally, this acetyl coenzyme A carboxylase, it is, you should know, it is the regulatory enzyme. So it will regulate the step of conversion of acetyl coenzyme A into the final product, that is your fatty acid. So finally, thank you everyone for your patience hearing. I wish you could have make a better sense of the process. If you like my video, just press this like icon. You can share this video with your colleagues as well as with your classmates, and for the further notification from my channel, you can press this bell icon. Thank you once again.